डाल दिया ना? हाँ, सब्जेक्ट से बोलो।
No, but the screen is not.
शेयर कर दो।
Okay, good. Oh, nobody didn't introduce a blank slide?
Position
Yeah, okay. No, that was fine.
is fine. Position
After this, yeah, after this.
this is
yeah.
blank slide.
This is blank slide, yes. Okay, so good. So, welcome. And last two lectures, I just want to, in one slide, I want to do a recap. Last two lectures, we discussed a great deal of the fundamental principles of DNA and RNA, nucleic acids, the basic elements of the nucleic acids we consider. And by and large, we say there are three kinds of DNAs or structures, A, B, and Z. These two are right-handed helices, duplexes. And this one is left-handed duplex, helix, left-handed helix. And all three are antiparallel strands. In every case, the two strands run in antiparallel fashion. And in the case of A and B, there is a mononucleotide repeat. And here it is a dinucleotide repeat. So what that means is that in every nucleotide here in the A and B, the same torsion angles repeat. In the case of the ZDNA, which is the left-handed DNA, the dinucleotide repeat means each of these nucleotides within this dinucleotide repeat will have different conformations. One of them has a C3' endosugar geometry, other one has a C2' endogeometry. Similarly, the glycosidic torsion angle, one of them is anti, other one is syn. And in the case of A and B structures, all are anti-glycosidic torsion angles. We discussed a whole lot of combinations there and various kinds of structures that come about as a result of this. And we also discussed a number of hydrogen bonding schemes. Watson-Crick scheme is only one of those, but there are many other possibilities of hydrogen bonding between the bases. There can be pyrimidine, homopyrimidine base pairs, and heteropurine base pairs, heteropyrimidine base pairs. All of these things we discussed in great detail last time. I'm not going to repeat all of that. Just a recap of all of those what we considered and the different structures that are possible with regard to that. And we are going to continue in the same direction now for a few more slides. And then we will, we also talked about the triplex formation, that how a third helix can come into the major groove of the duplex and bond, hydrogen bond with the two duplex. And the next possibility is the so-called quadruplex. Quadruplex is this called the G. By and large, it is observed in G tetrad. When there are 4 Gs, then they pair in the fashion which is indicated here. There are 4 G where symmetrically placed, and they are enrolled in the hydrogen bonding scheme, which uses up all the nitrogens and all the hydrogens in the G and the G base. Okay, so very symmetrical structure. You have a center here, and these are the 4 strands: 1, 2, 3, and 4. And that is, there are various ways one can form these 4 strands. They can all be parallel, as in this case. This is all parallel here. And here it is 2 are in one direction, 2 others are in another direction. These are individual strands which are going separate. Okay, all of these are G's, by the way. The center, this thing what you're saying is the G tetrad. This is the G tetrad. What is shown in this central plane, which is, that is the G tetrad. So G tetrads are stacking one above the other in every case. Okay, depending upon whether the chains run parallel or anti-parallel, there can be variations in the glycosidic torsion angles and the backbone torsion angles. Now, it is also possible that in these 2 cases, the top 2 cases, there are 4 separate nucleotides which are indicated there. But in this one, see, there are two nucleotides. They are going in this direction. One of them is going like this, other one is going like that. So this is anti-parallel. It is this, similar to this, except that there is a loop connecting there in between. That means the two molecules, the chain runs like this and then takes a loop and then comes down here again. And the other one is the chain goes like this and then takes a loop and comes down again. And then you form, at this stage, 4 G's are paired with each other to form a tetrad. So these are two molecules are involved. You can also form such a thing from a single molecule also. So look at this here. So here is the 5' end of one, the same chain, and here is the 3' end of the same chain. So 3'. So the chain runs like this, takes a loop, comes down, then takes another loop here, goes up, and then comes down, and this is here. In this way, 4 G's come in the plane as indicated here, and that will form the quadruplex. So these are called as 4-stranded structures, but it can be formed within by folding of the same chain, by folding of one chain or two chains or independently coming from different ones. All such possibilities exist. And this is enormous diversity in the case of structures of DNA molecules. And in a large DNA which consists of billions of base pairs, there will be stretches of CG islands, the so-called CG islands, and there will be stretches of Gs, especially in the C3 and the ends of the chromosomes. There are what are called as telomere ends. The telomere ends have G-rich sequences, and those G-rich sequences can form various kinds of structures like this, the quadruplex structures. And the telomere end, which is G-rich, actually plays an important role in the so-called aging process. Okay, so therefore, and there the length of the single-stranded DNA, which is at the end of the chromosome, this actually folds in different ways, and where the G tetrads are formed because they are rich in Gs. So length of this single strand, which is present at the end of the telomere, that is supposed to control the aging process. As one starts growing older, the length of the telomere starts coming down. Okay, and therefore the cells do not grow further. Whereas in the cancerous cells, the length is not coming down. In cancerous cells, the length is retained. So therefore, that continues to grow. The cells continue to divide, and therefore that causes the disease. And therefore, the telomere ends have been the target for cancer control. That also has been, so therefore all of these are important from the point of view of understanding the mechanisms of the diseases and controlling the diseases, design of drugs for the diseases. So the structures of these are important to understand. So for a long time, of course, the Gs were considered as the only ones which can form the tetrads. But later in the late 90s or something like that, I don't remember the years here, it was found that you can also form a different kind of a quadruplex structure for the Cs, using the Cs. Now the C, there are two Cs here. One of the C is protonated at one place, therefore it becomes a C+. Typically this is seen at low pH. Low pH conditions are found in some parts of the body, and that C gets protonated. When that happens, it can form a hydrogen bonding scheme like this, CC+. And then you have a structure again for the interdigitated structure. You have two strands, the G's are forming a base pair here, the C's are forming a base pair. Okay, and then you have another strand which is slightly shifted in vertical direction and interdigitates. Okay, intercalates between this CC pair is lying between this CC pair and this CC pair. This is lying between this CC pair and this CC pair, like that. So this is, it stacks in between. One CC pair is here from one strand and one pair of two molecules. Other one is like this, and the other pair comes in between these two, and that is called as interdigitating. That's why it is called as I motif. So this sort of a structure is called as I motif. It can be by different portions of the Cs that the DNA chain is having. You can form them in vitro with the different molecules. You can also have the same thing happening by the folding of the single chain of the DNA, where there is a C-rich. The C-rich regions, wherever they are there, and if the pH is low, then such a kind of a structure can also be formed. So these are all special situations. Under special conditions, you can form structures of this type. So you can imagine how much is the variety in the DNA structure that is possible, which is largely unexplored. So therefore, in these situations, these various structural techniques can play a very important role in understanding this. Okay, now this is a space-filling model of the DNA quadruplex, and 4 strands are indicated by different colors. This is the G quadruplex. Okay, this is not I motif. This is the G quadruplex structure, and 4 strands are intertwined in this particular manner. And in between, in the center, are all the G tetrands. Okay, now the DNA protein complexes can also be formed. DNA protein complexes means the DNA duplex is there in the particular fashion here, and the protein can bind at the major groove in this here. The proteins can recognize certain base pairs here and they can bind at this place. The DNA protein complex. So in that context, the recognition context, the structural variations in the DNA become important. Where the hydrogen bonds are possible, where the functional groups are exposed, that thing becomes important. So the DNA can bend itself to accommodate the protein so that it can produce a stable structure. And you see here in this situation, the DNA is completely folded. It's going around in a circle like this, and it is surrounded by proteins all over. And in this situation, you see the duplex is formed a kind of a circle, and there are proteins inside there. Proteins inside all these sheets with the cylinders which are there. These are all helices, and you have different kinds of helical proteins which can be present inside this, and the DNA wraps around this. See, this sort of structures are seen in nucleosome particles. In the chromosome, you have these beads, the so-called nucleosome particles. In that one, the DNA wraps around the proteins. There are various kinds of proteins there. The histones, the so-called histones are the major ones. And there are other kinds of proteins also. Therefore, you can see how much the DNA can take different shapes, and the DNA structure becomes therefore very important to understand what is all happening. Okay. All right. Now let's look at the types of RNA. Oh, that was the DNA. Now the DNA, I said there are RNA, there are 3 types of RNAs. One is called the messenger RNA. Messenger RNA one, which is this comes as a result of the transcription. This is the output of the transcription process from the gene to mRNA. Gene is a portion of the DNA, and once it is transcribed, it produces a single-stranded DNA, and that is called as mRNA. Then you have another RNA which is called as the tRNA. This is a relatively well-defined small molecule. It is about 76 nucleotide units, and it has this kind of a structure. This is so-called as the carrier of the amino acids. And this transfer RNA, this plays an important role in carrying the amino acids to be synthesized in protein synthesis. Amino acids which are to be added on the protein chain, and this is the ribosome here. This is the ribosome. Ribosome also contains various kinds of RNA here, and the messenger RNA goes and sits inside the ribosome, and the transfer RNA also comes inside. That I think is probably there in the next slide. No, here. Okay, now this one is there here. We'll go back to that. So you have the DNA here. The DNA, when a particular portion of the DNA opens up as a duplex, and then this gets exposed. Let's say this is a particular kind of a gene, and this gets transcribed into the messenger RNA here. And this messenger RNA goes and sits inside this ribosome. These are 2 units of the ribosome. This is sitting inside the ribosome. Okay. Now there is a so-called genetic code. Genetic code means the 3 nucleotides represent 1 amino acid. Okay, so that is how the protein chain is defined. If you have a messenger RNA of 100 units, then of course it defines a certain sequence of the amino acids which are to come in that one, because the 3 nucleotide units define one particular amino acid. So therefore, in the DNA synthesis, what happens is the messenger RNA keeps sliding over from one end to the other end, and the transfer RNA comes and brings the particular amino acid, and there are different kinds of transfer RNAs. For each amino acid, there are 20 amino acids. For each amino acid, there is one transfer RNA. They are very specific. It contains the genetic code at one end of the sequence. See, this is the so-called transfer RNA structure. Transfer RNA has this sort of a structure over here. You have a stem here and another stem, the variable loop. There is a T loop here, the D loop. These are all things which are defined in the schematic way. Then here is a loop which is called as the anticodon loop. There is the one which contains information about which amino acid this transfer RNA carries. So depending upon this code here, suppose we have TAG. Suppose this is the code, the three amino acids which are indicated, this is the anticodon. This defines a particular kind of amino acid, and that amino acid gets attached at this end of the transfer RNA. And the loaded transfer RNA goes inside the ribosome and then attaches the amino acid to a growing polypeptide chain. So we'll come to that. Okay, so now here this is the schematic of the structure and this is the actual structure of the transfer RNA. So look at the way this is called as the L-shaped 3D structure of the RNA. This was determined by crystallography, and the loop is here. This plays a very important role in identifying this kind of an amino acid which has to be carried. So therefore there are as many RNAs, tRNAs as there are amino acids. Okay, so therefore each one carries a particular anticodon here. So the anticodon is the one which tells you what sort of an amino acid has to be present here. The codon-anticodon pairing. Okay, on the mRNA there is a codon. On the tRNA there is anticodon. Right. So what does that mean? Suppose I have on the mRNA, this is the sequence. Suppose this is the sequence of the mRNA. Then the corresponding anticodon will be A, T, and C. So this will be present on the transfer RNA. That will be present here, because then only it can go and pair. It will pair with this, and this. The pair formation will happen here. So therefore, and then the mRNA slides in the ribosome. When the
For slides,
there is.
Last slide is 40.
No, I want to go on behind there. There was.
Which slide?
This one. Yeah. You see here, and the mRNA is sitting here. This will actually slide. It will slide once the particular amino acid is attached, and it will the new acid. Then this is the polypeptide chain which is getting synthesized here. So one by one, the amino acids are getting attached by the transfer RNA in the ribosome, and the mRNA keeps moving. And as amino acid is attached, the polypeptide chain starts coming out from the other end here. So this is how the polypeptide chain grows in this one. So DNA synthesis happens in the ribosome in this manner. And this DNA, as I said, this is one particular gene. Now another gene has to be expressed. Some other portion of the DNA will unwind. When it unwinds, it produces the single-stranded DNA, single-stranded RNA here, and that is the carrier of the information for the protein sequence. And there is an enzyme called RNA polymerase involved in this process of transcription. This RNA polymerase reads the sequence of nucleotides in the DNA, synthesizes the RNA corresponding to that, and that is the mRNA. Okay, the process of transcription, then there is the process of translation. So from mRNA inside the ribosome, this is the ribosome, and the ribosome is a factory. Is a factory where the proteins are synthesized. This is quite a involved process, and the discovery of this actually, some of you may know, led to the Nobel Prize to 3 people, and one of them is an Indian there, Venki Ramakrishnan, who is in UK now. So he was the winner of the Nobel Prize for discovering this process of protein synthesis, especially with regard to the antibiotics. The antibiotics, what they do is antibiotics go and bind at the particular site in the ribosome and block the mRNA and the peptide protein synthesis. And that is how the effect, the mechanism of action of the antibiotics was demonstrated by him in this process. So therefore, this understanding the structures, it plays an important role in understanding the functions, how the various drugs are functioning. Here the role of the antibiotics, how they stop protein synthesis, unwanted protein synthesis, that was explained by this, and that actually led to this Nobel Prize. Okay, now this is explained, the same thing is explained in a little bit more schematic detail. So here we have the messenger RNA coming in there. This is the DNA. From the DNA, the messenger RNA is synthesized. So this is here, and you can see here there are the codes. See, the different colors represent different kind of bases. Okay, so you have different kinds of bases, A, T, G, C. Okay, 3 nucleotides constitute the genetic code. It represents 1 amino acid. The mRNA comes and sits inside here in the ribosome. This is the ribosome, and the transferred RNA brings the particular amino acid according to the code which is here. The code which is UCG on the anticodon on the mRNA. That is a code. The corresponding anticodon is AGC. Okay, this I mentioned to you earlier. AGC, that is present in the transfer RNA here at this point. Okay, this comes and sits here. Once, and the amino acid which is synthesized is coming out from one particular end. Okay, growing peptide chain here as it grows. And once the amino acid is attached, this mRNA moves away from that site, and the new transfer RNA comes corresponding to the next amino acid. And that moves, the next amino acid will come. The transfer RNA keeps moving in and out, in and out, in and out. So this is a very complex, very concerted mechanism of protein synthesis. So it's very important to understand this. So many proteins play roles in this. And the attachment of the peptide, the chain is growing here, is done by an enzyme called as amino acid synthase. Okay, that enzyme comes, attaches here, keeps attaching one after the other amino acids as the chain is growing like this, growing peptide chain. Say tyrosine, glutamate, serine, alanine, tryptophan. So it keeps growing in that manner. Okay, so that is a schematic of how this protein synthesis works. So you see how much in knowledge we have gained by understanding the structures of these RNA and DNA molecules. Okay, so now this is with regard to the structures. Now we will go over to the NMR analysis of these nucleic acids.
So second slide.
Second PPT. Okay, so now we are all set to use NMR to understand DNA, RNA structures and the interactions. And here, because all this background was necessary to be able to understand the NMR analysis. The crystallographic analysis or NMR analysis. NMR analysis deals with the things in solution and individual atom level details one can obtain with regard to the interactions. It will also allow you to see how the dynamics happens in the different DNA molecules or the RNA molecules, how these things happen. Therefore, and here we talk about mostly proton NMR. Occasionally we talk about nitrogen 15 NMR or C13 NMR, but that will require labeling of the DNA at nitrogen positions or C13 positions. But most of the developments which have happened have been done using proton NMR quite successfully. So therefore, we are now ready to look at the DNA structure in terms of the proton NMR spectra. Not only 1Ds, 2Ds, 3Ds, everything can be done with this kind of things. So first thing to start is to see what are the chemical shifts. We have seen various kinds of protons in the DNA structure, right? We have seen the protons in the sugar rings. The sugar ring protons are here. These are the protons in the sugar ring. We said, remember the nomenclature now. I said, because we are going to use these nomenclatures of the atoms. So that is the crucial thing for so far as the NMR analysis is concerned. Okay, so here we have 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 5 double prime. These are the hydrogens. And in the case of RNA, they have a particular range of chemical shifts. See, the H1 prime is always about around 5 ppm, and the 2 prime, 3 prime, 4, they are come in this order, in this sequence there. If you look at the DNA, there is a change there. In the change, H1 prime is at the same place, but the H2 prime and H2 double prime, there is one more proton there, H2 double prime. Because in the case of RNA, in the ribose ring, that second one is an oxygen there. Therefore, you will not see. But here in DNA, at the 2' position, C2', there are 2 hydrogens. And therefore, that becomes H2', H2 double prime. Now, since there is no oxygen there, they actually get shifted more upfield. They come in this area. So H2', H2 double prime come in this area. Other things remain around the same place. So 3', 4', 5'. Okay, and the base protons and these are the amino protons. All of these have the base and the amino protons. Right, amino protons and the base protons, they all come in this area here from around 7 to 8 ppm, 8.5. So you have the base protons in this. The nomenclature is also indicated here. It is the same in both RNA and the DNA. H2, H8, H6, H2, H8, H6. And where are the H2s? H2s are present only on A. H8 is present on A and G, and H6 is present on C. Okay, there is also H5. H5 overlaps with the H1 prime area, and that is from the base area. From the base proton of H5, they comes with overlaps with the H1 prime here. So this comes H5 also comes in this area. This is present in cytosine. Okay, cytosine and uracil, H5 is present there. Okay, that proton also comes in this area. And then here you have the loops. Okay, the immunoprotons, the NH of G, T and U, they come in this range from 10 ppm to 15 ppm. Okay, the loops come in this area. We have seen various kinds of loops, because the loops meaning they are not hydrogen bonded. They are exposed, and when they are slowly exchanging, you will see those peaks there. So these are the loops. And then the base pairs appear here, the GC, AU, AT. These base pairs appear in this area. Which protons do we monitor here? We monitor the NH protons. These are the immunoprotons. We monitor the immunoprotons here exclusively. For that, one has to record the spectra in H2O. You cannot record in D2O. Okay, one has to record spectra in H2O to observe the signals here. Okay, and all this becomes very much dependent on the pH. What pH you do, because the pH controls the exchange rate. pH of the solution, how much is H3O plus inside in the solution, they exchange with the NH. These are exchangeable protons. NH and NH2, these are exchangeable protons. NH2 is generally exchange much faster, and you often don't see the NH2 protons. But if there is a lower pH, you might see them also. NH2 protons also you can see. Sometimes in a very strong hydrogen bonded network, like in the G quadruplex and triplexes, you may, which are hydrogen bonded aminos, then their exchange rates are reduced, and you will see them also. NH2s also. Therefore, all of these things one has to remember to analyze the NMR spectra of the nucleotide units. Okay, this is the typical proton NMR spectrum of non-exchangeable protons. This is the sequence which is taken here. This is how many residues here. So this is 6, 8, 6, 14 nucleotide units. Okay, now is this what kind of a sequence it is? Now suppose, can it form a duplex? The structure, the sequence, if you look at it, can it form a duplex? If a particular sequence form a duplex by itself, then it is called as the self-complementary sequence. Because if you write that in the opposite direction, suppose I have a sequence, I write here GATC. Okay, this is the same molecule. I write the same thing in the opposite direction because you have antiparallel duplexes, GATC. If I write it, then you can form base pairs here. So this is 5', 3', 5', 3'. Two molecules of the same sequence, they will base pair and they form a duplex. So such a sequence is called as the self-complementary sequence. Okay, so it can form a duplex, very stable duplex. And here you see, is it a self-complementary duplex? Not entirely. See, if you look at the first 6 units here and the first 6 units here, these here, they are the same. So therefore, if I write in the in the opposite direction, this portion will form a duplex, this portion will form a duplex also. But the central 2 C's, they don't pair. Therefore, it will loop out. So those C's will loop out of the duplex. So it will form a structure which may be something like this. Okay, it can form a structure something like that. or it can also form a structure like a hairpin. Okay, this 6 and this 6, the two ends can stay here, and or if there are two molecules involved, then it can form a structure like this. Now, a typical NMR spectrum of this is the non-exchangeable protons. In the non-exchangeable protons, what are we going to see? We will not see the iminoprotons and the aminoprotons. We will only see the sugar protons and the base protons, and they are indicated here. The methyls appear in this region. H2 prime, H2 double prime appear in this as I indicated before, and the 4 prime, 5 prime, 5 double prime, they appear in this area. 3 prime appears here. 1 prime and H5. H5 is from cytosine. This will appear cytosine or uracil in both cases. Uracil in the case of RNA and cytosine in the case of DNA. And H8, H2, H6, they will all appear in this area from 7 to 8 ppm. Okay, so that is indicated in this here. Okay, and then you can see here, this is another sequence, and this is what we am showing here is the iminoprotons. Iminoproton, the sequence is like this. You have the GATC, TT, CC, CCCC, and GGAA. That is the sequence. Okay, so now what I will do is, what sort of a structure it can form, the stable structure it can form. So I will start writing GATC, we have a GATC here, okay. GAT is the starting point. Now I will start the second molecule, I will start writing from here, GATC, then TT, CC, see, so much of duplex formation has happened, all the way from here to here. And if I make a loop of 4 C's here, 4 C's here, although there is a break at this point, there is no connection from here to here, but it can form a very stable structure because this so much of base pairing has happened, Watson-Crick base pairing here, chains running in the anti-parallel direction, and they will form base pairs. And even though there is no connection from here to here, the stacking of this TA base pair and the CG base pair here, it provides a good stability to the system, and it can form a stable structure. Now you see, therefore, there is a symmetrical duplex formed of 12 nucleotide units. How much? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So the 12-mer duplex gets formed in the middle, and there is a loop on one side of either side of 4 C's. Okay, now how do we know this? So if this is the kind of a structure which is getting formed, then I should be able to see the iminoprotons of the base pairs. So which are the iminoprotons we will see? The G's have the iminoprotons and the T's have the iminoprotons, right? So therefore the G is here, T is here, and the two T's are here. Okay, I will see this. And then I will also see this G and this G. This G, these two T's, this G and this T. And this is symmetrical. Exactly the same thing appears from the other chain also. Therefore, I will see these many iminoprotons. How many? These are 6. The 6 protons, 1, 2, 3, 4, 5 and 6. And those ones you can see, you can count here, 6 iminoprotons are seen. Okay, and this is at 274 degree Kelvin at 1 degree centigrade, because the iminoprotons exchange rapidly. So if you want to observe these iminoprotons, you have to do this experiment at very low temperature. And here it is indicated the AT base pair and the GC base pair. And the GC base pair, what we are observing, AT base pair, what we are seeing is this iminoproton, and the GC base pair, what you are observing is this iminoproton. The aminoprotons which are which are present here, they generally are not seen. They exchange quite rapidly, especially when you are at neutral pH, around 7 pH. Often you don't see this. So you will see the iminoprotons only. So these protons, they are very strong and very stable, and you can see 274 degree Kelvin, 278, 283, 288, 293. As you increase the temperature, the stability of this duplex keeps going down. And therefore, when that happens, the iminoproton starts getting, starts exchanging, and then the lines start getting broadened, and eventually they will disappear. At 303 degree Kelvin, that is 30 degrees centigrade, you see this structure has broken down. The stability has gone down. So if from here, you look at the 274 degree, you can count the six peaks here, 1, 2, 3, 4, 5, 6. There are 6, which clearly tells you that I am forming a structure of this type at this low temperature, symmetrical. Okay, so the immunoprotons provide you a first-hand information about the nature of the duplex that is getting formed. Okay, a similar thing, the DNA hairpin thermal stability here, and you see in this particular case, you see this G, this is the similar thing here, and you see the T's here, okay, T's there, you will see those peaks there. And then you have the XN, this is the loop area here, this is the single molecule here. This is the single molecule turning around. So this portion is self-complementary, GAATTC, GAATTC, and in between you have a loop. This is called as the hairpin structure. When you have the hairpin structure, this loops out, and this portion of it forms a base pair with all of these other ones there. So it forms a stable structure. So how many you should see here? You should see 6. Okay, and you do see 6. At low temperature, you do see six of those. This is 5 degrees, 25 degrees, and 35 degrees. Okay, these ones are the immunoprotons, and these ones are the loop protons, which I indicated there earlier. What is present in the loops, we do not know. These are T loops. T, this is in the area that is seen there. Some of those ones are seen in these loop areas. So these are the base paired ones. As we increase the temperature, they will start disappearing. Now this will also tell you, it will help you in the assignment. You monitor the changes in the intensities. This will tell you which of these base pairs are opening up first. Because typically, which are on the exterior ones will start opening up first. Okay, as the endpoints are fraying. Therefore, they are not so stable. That is the one which first opens, and then the next one, and the next one, and the next one. So that's how sequentially they start opening. This is called as the melting of the DNA. Melting of the DNA duplex, the hairpin. So you can see here in these 6 peaks which are there, this is the weakest. The weakest one is the weakest, and that belongs to the first base pair. That is weakest because it is exchanging with the water. Intensity therefore is low. And increase the temperature, you can see that is the first fellow to go. Okay, that's the first fellow to go. And after that, the things will disappear, or you can see these ones slowly. One of them, this T, this T goes next. This 2, this is number 2. This is number 1. You monitor this is number 2. Therefore, you identify that as number 2. And which goes next? After that, you identify which one goes next. Then you can figure out that, okay, that is number 3 here, then number 4 here, 5 here, and this is the last one to go, that is 6. So this is in the interior. So that is how you identify the individual resonances on the basis of the melting sequence of the individual immunoproton resonances. So this is a very useful information from the DNA structure analysis. Okay, so this is the melting of the duplex DNA. The same point is made here as well. So at 0 degrees, you have all of these immunoprotons here. So you have so many 6 immunoprotons there. And as you start in, because there is a symmetrical DNA. This is the 12-mer symmetrical DNA, which means there are only 6 G's and the 6. Together you have G's and T's, you have only 6 of them. So you have 1, 2, 3, 4, 5, and 6. So 6 T's and G's only are the immunoprotons, and what are present here are the G's, and what is present here are the T's. So therefore, by systematically monitoring the progression of the disappearances of the peaks, you can identify the individual immuno peaks as belonging to particular nucleotide unit along the sequence. So that is what is done in this case. At 60 degrees, this is more or less completely broadened out. So you can see here, whereas CG 1, 2, 3, 4, and 5, and 6. This is how you identify the individual. This is called as the melting of the duplex DNA. That was what was done in the very initial days of NMR, the structure determination of large DNA molecules, meaning you could study the melting of the DNA, stability of the DNA, and various kinds of things. You could study the effects of temperature, effects of modifications inside the DNA sequences, mutations if they happen, how they influence the things. These are all the modifications which are there. So we can skip this. Okay. So now we come to the duplex structure determination. Okay, that is we have seen how to identify the duplex. We have seen how the immunoprotons are telling you about the stability of the duplex, how it is opening up, and now to determine the finer details of the structure at atomic resolution, now we have to go to 2D NMR. The 2D NMR, we analyze the non-exchangeable protons and the contributions of non-exchangeable protons in the NMR spectroscopy is well recognized. That's what we are going to discuss now. How we use this to calculate the three-dimensional structure of the DNA at finer level of detail. Individual residue level, how much is the variation in that one? That is the thing which one can determine by using the standard 2D techniques which become available as we discussed in the previous set of lectures. So there are two fundamental experiments as we are just for a recap here, that COSY and the NOSY. COSY is a 2D experiment which will give you J correlations in the spectra, and the NOSY is an experiment which will provide you the NOE correlations or the distance correlations, the distance maps and the J maps. So these are the two things which are important, okay? And whether these techniques, whether you use for DNA or proteins, it is all the same. The methods are the same. So we can use this for DNA as well here. So now here is a typical NOSY spectrum of a 14-mer DNA sequence. For ready reference, I put these ones also here. Oops. For ready reference, I put the primary structure of the DNA as well as some details about the distances between the protons indicated here in the bottom one here. So here I show you some of the distance information. This is what is obtainable from the NOSY. Okay, you have this I here and I+1 nucleotide unit, and the base of this nucleotide unit is this, base of this nucleotide unit is this. So here what I have an A here. This is the A nucleotide because I have a H8 here. You see, H8 here is only there for it is there for both A and G, but H2 is present only in A. In the G at H2 position there is a NH2 group. Okay, now and then you have this sugar ring of one nucleotide unit. This is the sugar ring of the next nucleotide unit, and the phosphate of this one is here and the phosphate of this one is here, the CH2. Okay, so this is just a recap of what we discussed earlier. Now the 2D NOSY spectrum will show you correlations between within the same nucleotide unit, that is from the base proton to its own protons in the sugar ring, and it will also show correlations from base proton of one unit to the sugar ring protons of the next unit on the 5' side. Notice here, this is 5' to 3'. And we are going to see correlations from the base proton of this to the sugar protons of this nucleotide which is at I. This is I+1 and this is I. We will see those. We will see from here to here, here to here, and we also may see from here to here as well, the base to base. And that is indicated in this here. From this you will see correlations in the NOSY spectrum. They will have different intensities, and this intensity will depend upon the inverse 6th power of the distance between these proton pairs, and therefore the distances are also important. You have to see how much are the distance in these cases. So this distance is 3.5 angstroms, and you can also have a distance of from here to here it is 3.5, from here to here it is 4.3 angstrom, and accordingly the intensities of the peaks will also be different. So therefore you can monitor this to calculate the structure of the entire long DNA sequence. So here is the long DNA sequence. This is the 14-mer again, CG, CG, AG, TT, GT, CG, CG. See, now you see this is self-complementary? Suppose I want to write here CG, CG, A, CG, CG, A, and after that it is not up to here it is self-complementary. Okay, and these central 4 units, GT, TG, these ones do not form base pairs. Okay, therefore they form out a loop. They loop out of the duplex, and if I write the sequence in this manner again, either it can form a duplex or it can form a hairpin. Hairpin means the GGTG will loop out and forms a 4 base loop, and these 5 nucleotides and 5 nucleotides will form a base pair stem. So you have a stem and then it will form a loop. So it is something like this. So you will have this and you will have a loop, and in this loop you will have G, T, T, G, and others, these two form a base pair stem. So this sort of a structure which is present here, this create quite a bit of heterogeneity. Now you look at this spectrum, this is beautiful, all very beautiful. All the 14 nucleotide units can be separately identified here. There is so much dispersion among the various peaks. You see various kinds of cross peaks. You see cross peaks between the base protons here and the sugar protons here. The CH3 belongs to what? CH3 belongs to thymines, and the 2' 2" belong to the sugar ring. So you will have correlation from the base protons to the sugar protons here, and to the sugar protons here, from the methyl to all of these here, and also to the base protons here. Base proton. Then within the sugar ring you have these correlations here, 2' to cross peaks, and then you have these are the 1'. This is the 1' area, right? This is the 1' area. So you will have the 1' to the various sugar rings here, 1' to 3' and the 4' and the 5', and then to the 2' and the 2". These are from the 1'. Similarly, from the base protons here, you see within the base correlations here, base that is H8 or H6 or H5 H6 H5 will come in this area, and this area belongs to base to the H1' and to the H5. Then you have base to the H3', then the 4', 5', base to the 2' 2" here in this area, and then you have the base to the base, that is to the methyl. So therefore this is extremely informative. So much information is present. Quantitation of individual peaks here will provide you the individual distances as I indicated in this area. What is the distance between a base to base, one base to the next base? Notice here, of course, that you will not see a from this base to this base. That is a long distance. You will see, base to base you will see. You will not see from this base to this sugar protons. You will see from base to base, that you will see. You will not see from this base to this sugar protons. That is base of I to the sugar of I+1 you will not see. You will see from base of I+1 to the sugar of I, the previous nucleotide units. Therefore, there is a directionality in the NOEs which are present. From the base proton to the sugar protons, there is a directionality. Base to base, there is no directionality. So here you can see from each base, you will see two peaks, one following, one before. So you will see all the base to base peaks. So once you identify that, you know what is the sequence of the bases which are present in your DNA sequence. And from this base to the 1' and of two peaks you will see, base to 1' of its own sugar, base to 1' its own sugar, and base to 1' of the sugar on the next slide. Similarly, for these areas also you will see cross peaks from those ones. So a quantitative analysis of all of this is extremely critical for understanding the structure. Okay, now here is the list of all the short distances which are present in RNA and DNA. And why this is listed here? Because this is important to know what sort of peaks you expect in the NMR spectrum of the DNA sequence. And if there is any distance which is more than 5-6 angstroms, you will not see a cross peak there. Okay, so here you see for the different kinds of DNA sequences or structures, you have the short distances which are less than 5 angstroms, except one or two here, which is this is for the Z DNA. In the case of Z, for the A and B, all of these are, what are these ones here? H6 to H1 prime, H6 to H2 prime, H6 to, these are within the same nucleotide, H6 to H2 double prime, H6 to H3 prime. Now you see for the G, HA to H1 prime, HA to H2 prime, HA to H, these are all within the same nucleotide. Okay, these are intranucleotide distances. And now here you have the internucleotide distances. So this is N and N-1. If there is a CG step, you will see H6 to H8, HA to H6, H6 to H1 prime, H6 to H2 prime, H6 to H2 double prime, H6 to H3 prime, and so on and so forth. These are internucleotide distances here, and these are intranucleotide distances. Okay, so these sequential distances are present only in a sequential manner. Notice here N to N-1. Notice, remember this is N, therefore you have a directionality. You don't have N to N+1 with regard to this here. Only with regard to the base you do have that, but not with regard to the sugar. Okay, so that is indicated in this one. So you have to use all of these things for assigning the individual resonances in your spectrum. Here is an example. Here's an example of a particular portion of the spectrum. Which is this portion? This is H8, H6 to H1 prime region. What is that one? Let me show you that once more. And that is this portion. Okay, that portion that is indicated here. So because you see here, this is the base and this is the H1 prime area. The sequence is given on the top here, TGGC, GGG, T, and this is in solution sodium plus. And what is the terminal sequence? Terminal sequence is T8. Now T8, the green and the red, they indicate the self and the sequential peaks. Okay, self peak is in one color, sequential peak is in another color. Okay, now T8 shows this is the T8 position. How do I know this is the T8 position? Because this is, you see, also see from the methyl. T8 has a methyl. The methyl to its own H1 prime, methyl to this H6 proton, you will see these are the H6 proton you see. H6 and H8. So this identify the T8 position. Okay. You can also identify the T1 position. T1 position, how is it distinct compared to everybody else? Why? Because there is nothing above 1. Therefore, this fellow gives you only one peak. It doesn't give you the second peak. Whereas every other one which has something on the 5' end will produce a peak. Therefore, every other nucleotide has two peaks, whereas the T1 has only one peak in this direction. Okay, so therefore you identify this peak as T1 straight away because there is nothing else. From the T1 you go horizontally, you identify the base which is producing a sequential peak to this. Sequential peak to T1 means it is the next nucleotide, therefore it must be G2. T1, if it is this must be G2. And the G2, this is the G2 to T1 sequential peak. And what is this peak, green one? This is the self peak of G2. So G2 H8 to its own 1'. Now, if you go horizontally from here, you come to this place. This gives you the sequential peak of the next nucleotide unit to the H1' of G2. So therefore, this must be G3. From G3, this is the sequential peak. What is its self peak? Self peak appears here only. It turns out that these two peaks are overlapping. Therefore, half of this because this one comes at the bottom end of this peaks and the top end of this peak is separate. And you see this is the self peak of G3. And then you go horizontally from here, this comes in line with this peak. Therefore, this is the sequential peak of C4 and to G3. So this is the sequential peaks. The self peak is here. This is C4. And from C4, you go horizontally again. Then you get the sequential peak of the next nucleotide unit, that is G5. So G5 to C4, this is the sequential peak and this is the self peak. Now again horizontally you go, you get the next sequential peak, that is G6 to G5, and the self peak is here, G6. And once more, G6, you go to G7, this is the sequential peak and this is the self peak of G7, and G7 you go to this, then you go to the T8, T8 self peak is here. So that completes the assignment, right? So this is how you identify the entire set of assignments of the individual base protons and the sugar proton, the sugar H1' proton. Once you know the H1' proton, then you go here. You identify the 2' and 2 double primes. Or you can also go here. From the base protons, you identify the 2' and the 2 double prime. Because the H1' primes you clearly know now sequentially. Sequence-wise you know. Therefore, correspondingly, what is this? You can also see that in the Cozy spectrum. In the Cozy spectrum, it will show you intranucleotide cross peaks. 1' to 2', 1' to 2 double prime, and so on. But by and large, that is also reflected in the Nosy spectrum. So here itself we can see. And the 2' and 2 double primes, they are showing cross peaks here. The 2' to double prime correlate because there is a short distance. This is only 1.7 angstroms, the 2' to double prime. This is geminal protons. Therefore, you will see the cross peaks here. And that will match with this. If I take any of these two here, the corresponding peaks will indicate these are the 2' and the 2 double prime peaks. Okay, so this is how we obtain the assignments. And next, of course, we look at the sugar ring. We discussed in detail the sugar ring, what all the features are there: 3' endo, 2' endo geometry, sugar puckering, and this also we discussed last time. I will skip this, otherwise this will be quite repetitive of this. Only thing we want to notice here are the coupling constants. What are the kinds of coupling constants what we have here? In the case of ribose, the three bond coupling 1' to 2' is approximately 1 Hertz. This is angle is 90 degrees. This is approximately 1 Hertz in the C3' endo geometry. In the case of deoxyribose, then the P, this is in the S region, right? This is the C2' endo geometry. In the C2' endo geometry, 1', 2' coupling is 7.9 Hertz, and 1' to double prime coupling is 10 Hertz. These are large couplings. Therefore, you will see these cross peaks very well. You can see that here. Here is a plot of the coupling constants, all the 5 coupling constants in the sugar ring. Which are the 5 coupling constants in the sugar ring? You have 1', 2 double prime, 1', 2', 2', 3', 2 double prime, 3', 3', 4'. These many 3 bond couplings we have there. And all of these will vary depending upon the pseudo rotation angle. Pseudo rotation angle as it goes from 0 to 360. Okay, and all of them have different behaviors with respect to the pseudo rotation angle. Depending upon what is the sugar geometry, they have different coupling constants. Okay, so therefore you see in very critically you can see for this one here, this is the 1', 2'. 1', 2' has a maximum value at 144. P is equal to 144. And what is the P is equal to 144? This is the C2' endo geometry. So therefore in the DNA, this coupling constant will be very large. Okay, and in the RNA, 1', 2' coupling, and RNA, what is the P value? RNA, the P value from C3' endo geometry, the P value is here, and therefore that will be more or less 0. Okay, now similarly, you look at these ones here. These you have the 2 double prime 3', 2 double prime 3', and 3', 4'. In the C2' endo geometry, that is in this area, both these couplings are very small. 2 double prime 3' and 3', 4' couplings are very, very small. Therefore, you will not see these cross peaks. Whereas these two couplings, these are the 1' to double prime and 2', 3', they vary in the range of 6 to 8 Hertz or 6 to 10 Hertz, and you will see those cross peaks in every sugar geometry. Whether it is 3 endo or 2 endo or anything else, you will see those values because values are more than 6. Therefore, these become very crucial to identify what kind of a sugar geometry you might have in your DNA sequence. Okay, now this is a typical two-dimensional spectrum of a single nucleotide, 2' endo sugar. Okay, what are the peaks there? So this is the 1' to 2' to double prime, and this is the which base is this? So here you have a peak, and this is the 2' to double prime cross peak. Okay, and this is the 3' here, 2 double prime to 3, 2' to 3' you have here. As I said, 2' to 3' you will see here in the case of C2' endo. You will not see that in the case of C3' endo. So 2', 3' you are seeing here, and you see 2' to double prime. This is all you will see in the case of a C2' endo sugar in the H1', H2', H2 double prime, H3' region. So this is the 3' here, this is the 1', these are 2' and 2 double prime. So I should change the color of this, I guess. Okay, now you see black, right. Okay, so this is the 1', this is the 1', this is 3', this is 2 double prime, and this is 2'. So you will see cross peaks between these two for a single nucleotide, single sugar. And now, of course, as I said, depending upon the p value, all of these things become very complex. And to understand all of these things, you need to do simulations. There are lots of simulations which have been done, and I'm showing you the peak patterns of these various cross peaks for different values of p. Values of P's are indicated here, 9, 36, 72, 90. Okay, here is the 3' endo region, this is the north region, and on this side is the south region, the 2' endo region here. So you can see how the peak intensities vary. Peak patterns and the peak intensities vary. This is a region of H1 prime, H2 prime. This is in the Cozy spectrum. And this is the H1 prime, H2 double prime. H1 prime, H2 double prime peak. If the simulations indicate the patterns of the peaks, fine structures in the individual peaks. So, okay, now, should we take a break here?
Yeah.
Yeah, so 1 hour is,
Yeah, break.
we can break for 5 minutes, yeah.
You write that 5 minutes break.
Okay. So we are, I'm showing you here the various kinds of patterns you will get in these various cross peaks in the cozy spectrum, depending upon the sugar geometry. This is the 1' to double prime here. You can see quite a substantial variation in the fine structures of the individual peaks and the intensities of the various components here. This is because of the cancellations. Cancellations will happen depending on the coupling constants. And the coupling constants are varying, as I showed in the previous slide there. So therefore, you will have a very slim peak for the 1' to double prime in the case of C3' endo, but a wide peak in the case of C2' endo geometry here. So that is how the patterns vary in the 1', 2', and 2 double prime area. And this is another experiment which is also cozy, but it actually is not a cozy 90, but a cozy 45. The last pulse in the case of doing the 2 pulse experiments can be 45-degree pulse, or it also can happen in the case of what is called as the E-cozy experiment, which we also described in the previous set of lectures. And here the number of components is reduced by half, and you can see a certain kind of an orientation. The patterns will slightly change. Okay, and this will also be very useful to identify what sort of a sugar geometry you have in your molecule. If we don't see these peaks at all, then it means it is in the C3' geometry. But you see strong peaks here in this 162, 180 strong peaks. It indicates you have sugar geometry in the area of 162, 180 and so on. And that is in the C2' geometry, typically for the BDNA. And this is for the 1' to double prime peaks, where the resolution is much better because the coupling constant is quite more suitable. And the 2 double prime 3' peak has a smaller coupling constant, and therefore that doesn't show up as much. And whereas the 2', 3' shows up always. And therefore the fine structure is much more clearer here because of the fewer coupling constants. And you see this peak has a very different structure here. This is a C3' endo is a very different structure compared to the in the C2' endo sugar geometry here. So by looking at this, you can actually measure the coupling constants also. By identifying these individual things and simulating these peaks, you can determine the coupling constants as well. So there are lots of simulations here. We will not go into the individual details, but anybody who is actually working on it can refer to this to figure out where his sugar geometry is in your particular DNA molecule. Okay, so this is another one of the same. This is the 2', 3' coupling constant, 2', 3' cross peak. Once again, you see a lot of variation in the peak pattern as you go in, as you vary the sugar geometry. All from here to here, there is so much of a variation in the cross peak fine structure, 2', 3' peak. Okay, so therefore you can use this to identify the sugar geometry in your molecule at a very high resolution. So this is once more a similar one, except now it is a 45-degree or the E-cozy spectrum. In the E-cozy spectrum also you will have different patterns and you can identify. So this is for record. You have these ones already published at various places, and you can use this material whenever you would actually analyze the spectrum of a DNA molecule. Now here is the full spectrum of this same sequence which I showed you earlier. This is the E-cozy spectrum. You can see the very beautiful peak patterns here which is shown by simulation which I showed earlier. The advantage of that is that many of these peaks which are overlapping can be resolved by making this E-cozy. Of course, one can further resolve it by doing constant time cozy experiments where you remove the coupling information from one side, and that will allow you to measure the coupling constants better and resolve the peaks. So here you see how to measure the coupling constants is indicated here. In a cozy 45 experiment, we have seen one particular cross peak, 2 cross peaks with regard to one particular sugar H1 prime. This is the H1 prime H2 double prime peak, and this is the H1 prime H2 prime peak. And what are the coupling constants there? So far as the H1 prime is concerned, H1 prime has H2 prime and H2 double prime. Okay. Now this one has a coupling to H3 prime and to H2 double prime. This one has, of course, a coupling to the H3 prime and H2 prime. Nice. So therefore, these fine structures will appear here. The 2' fine structure will appear on this axis, 2 double prime fine structure will appear on this axis, and the 2' to double prime coupling will be present in both cases. And those are therefore 3 coupling constants are there. So you have the 2' to double prime coupling is 14.1 hertz. That is the geminal coupling. That is easily identified from this, the endpoint here to here, and this gives you the 1', 2' coupling, which is about 1 to double prime coupling. 5.8 hertz is a 1' to double prime. So here you have, this is 1', and 9.5 hertz is 1', 2'. This is the 2, and then 14.1 hertz is present in both cases, in all of these cases. Okay, so from here you can determine the coupling constants. Once you know what peak we are talking about, what peak you are talking about, you can figure out what are the coupling constants which are present, and you can measure them. Okay, so this is the simulations once more. Now you can see for a smaller molecule, TGGCCA, mitramycin, and this is the larger molecule, and this is the experimental spectrum, and this is the simulated spectrum. When you match, overlay this one, you can actually determine the coupling constants very quantitatively. This is for one particular nucleotide unit which is shown, but you do this for all the nucleotide units. Okay, so I've just shown you the simulation. In fact, this is the work which is, this is from our own lab. So this is in this large molecule. You can see the beautiful simulation which is matching almost entirely with this. Even in a complex structure which is like this, you can simulate it and match the individual peak patterns. Now, these are the things in the constant time cozy. In the constant time cozy, you remove the coupling constant along the F1 dimension. Along this dimension, you remove the coupling constant. Therefore, the pattern gets quite simplified. You have the coupling constant present only along the F2 dimension, that is this dimension here, and you can measure this coupling constant directly. This simplifies the spectra quite substantially. See, again, I have shown the same things here. You can also determine the H3' peak coupling, H3' phosphorus coupling. H3' is coupled to the phosphorus also. Okay, so therefore that coupling constant also will show up in the, when you look at the fine structure of H3'. In the H3', you will have that, so that will appear along this axis. It will appear like that. Okay, so you have to include that for simulation. When you simulate it, you can include that and measure compare with the experimental spectra. Now you see here, this is an interesting example. This is the same molecule which is shown here, and it's the E-cozy spectrum. E-cozy spectrum was this. This is the E-cozy spectrum. This is the E-cozy spectrum of this molecule. Look at the complexity which is present here in this area. Of course, we simulated this and analyzed it, but can we make it further simpler? Okay, that is what is done by the constant time cozy. In the constant time cozy, you saw this here. See, in this area, there was so much of overlap of peaks. All this was overlapping. This was overlapping. You could not figure out which are the ones nucleotides which are present here. Here you can clearly see there are 2 nucleotide units present here. You can identify them. They are slightly shifted, one from the other to the left. Okay, this can be clearly identified in the constant time cozy experiment. In the case of normal thing, these were all overlapping in one together, and these are also overlapping. Okay, in the E-cozy spectrum, one couldn't figure out that. But now here you can see them very clearly. That separated out because you have removed the information along one axis, coupling information along one axis. And in this you can identify the pairs also clearly. This goes with this, this goes with this, this goes with this, and this is the blow up of this, showing the fine structures. There are also positive and negative combinations here. Okay, so that is important. Although it is not showing up, it is not a color picture, but there will be positive and negative combinations there. Okay, this is the 2', 3', H2', H2 double prime, 3' area. This is impossible to analyze in the case of normal E-cozy or the cozy spectrum. But in the constant time cozy spectrum, this sort of a situation can be easily identified. Look at this, they are getting resolved here. They're differently placed. The 3' chemical shifts of these 3 are different, very close. But if you have the coupling information here, they will not be able to resolve this at all. But by decoupling along the H3', you are able to measure this, separate them, and you can actually measure the coupling constants. Okay, so these are the simulations of the same. We'll skip that. Now this is with regard to the backbone. What is the possible torsion angle epsilon? So you have the energies, and all the torsion angles are they allowed from 0 to 360. So this is a energy calculation has been done here to see the possibilities of various values for the epsilon torsion angle. It turns out that epsilon is equal to 180 or is equal to 270. This is the minimum energy conformation. For others, the energies are very high. So it is very unlikely that you will populate any of these values. So mostly you will either have 180 or 270. These two possibilities are existing, and mostly you will see into BDNA, you will see it is all here. This is also seen in the case of the crystal structure, which I showed you earlier, and also in the NMR structures, this is also shown there like that. Now this is the backbone, another illustration of the H1 prime H3 prime area. H1 prime H3 prime area has all this fine structure. You can calculate the P31 coupling constant from here. H3 prime P coupling constant determines the epsilon torsion angle. Why is it important to get the H3 prime P coupling? Because it reflects about the epsilon torsion angle. H5 prime, H5 double prime to the phosphorus coupling constants, this will tell you about the beta torsion angle. Beta torsion angle, where are they? These are in the backbone, right? Alpha, beta, gamma, delta, epsilon, xi. So these are the torsion angles along the backbone. Now if you measure these coupling constants, H3 prime P and H5 prime, H5 double prime P, these will tell you about the epsilon and the beta torsion angles. That information is present here in this spectra. Now you can simulate this to calculate, to obtain this coupling information. Okay, now this was the technique which we developed. Of course, I am not going to discuss that in detail here, but this paper is already there. So to measure the phosphorus proton coupling constants from in oligonucleotides. Okay, so like we got now the coupling information. We also, from the NOE, the noisy spectrum which I showed you earlier, you can also quantify those intensities of the peaks and calculate the distances. The distances, because I showed you the distance information for the various peaks. Okay, you can calculate the approximate distances and say, okay, this particular kind of proton pair has a distance within this particular range. So it has the lower limit and an upper limit. So on the basis of the intensity, you define an upper limit, that is here, and a lower limit, which is here. And so any model you build, any structure you determine must satisfy these limits. In other words, it should be within those, in between those two limits. Okay. Then what you do is you do it automatically on the computer. If you want to do it, you kind of define an energy function, which is E is equal to EF plus ENOE. EF is the energy coming from all the various other kinds of internal bond angle, bond distance, all variations, violations, and ENOE is the thing which is coming from here. You define the ENOE in this manner that if there is a deviation from the lower limit, you put this constant here. This is the force constant in this. Like a harmonic oscillator, this is a potential function for violation in the lower limit. This is the harmonic function for violations in the upper limit. So you, for every peak, for every proton pair, you calculate this and determine the sum of all of this. Every proton pair meaning what? You build a model. You start with a particular DNA model. Suppose you have a 10-mer DNA model. You have so many proton pairs. Every nucleotide. So for all of those proton pairs, you calculate the distances from your intensity patterns and see whether, and you define the approximate limits from the intensity patterns in the noisy spectra. Then in the model, you calculate the distance, what is that particular proton pair distance, and you see whether it lies within that range. And to quantify that, you define this potential function. And then you keep optimizing this model. Keep optimizing the model until this condition is satisfied for both the upper limits and the lower limits. So if that is satisfied, then you get a value 0. If the restraint is satisfied, you put a value 0. Okay, if it is not satisfied, then this has this kind of an energy contribution. If it is satisfied, you put a value 0. If it is not satisfied, then you have this contribution to the potential energy for the lower violation and the upper violation. Okay. So therefore, you optimize your model so that you get a 0 for this ENOE. Then it means that all your proton pairs are within the range that is expected from your noisy spectrum. So this is done by what is called as a distance geometry algorithm. This was developed by one of our students. It's called a torsion angle based distance geometry, TAND. It is called as distance geometry. So you start from an arbitrary model here like that for the DNA, a particular DNA sequence. This is a 12-mer DNA here. Arbitrary model, anything, there will be all kinds of clashes and this, that. Now you have this EF potential function which takes care of all the clashes, steric clashes. Then the NOE potential distance constraints you have. Now you optimize the torsion angles along the backbone and keep changing your model and calculating the ENOE. And you see, as you progress through the iterative procedure, eventually you will end up with a structure which is like this. See, now it is a proper acceptable structure. When all the conditions are satisfied, you are getting a structure which is like this. So which you see, naturally it is coming out as a right-handed helical structure. So this is called as a nucleic acid distance geometry algorithm. These are all incorporated in all these major structural biology models. Those ones are all there inside. Okay. So now this is getting into a little bit more complex situations. We will not spend too much time on this. I just give here for information. To here you make use of heteronuclear experiments as well. See what is present here, the carbon chemical shifts. These are carbon chemicals, C4 prime, C1 prime, okay. This is H1 prime, C1 prime, H4 prime, C4 prime, correlations. How do you get this? You get this from experiments called as HSQC, heteronuclear single quantum correlations, right? So you have this H1 prime, C1 prime. We have discussed this in the previous set of lectures. H4 prime, C4 prime, okay. And then you have this from the base also, TNH3 to nitrogen. These are nitrogen chemical shifts. You have the nitrogen chemical shifts, the carbon chemical shifts here, and you come for the various individual nuclear assignments are made. We will not go into all of those details. Just I'm indicating to you that you can also use heteronuclear experiments like HSQC to identify the individual carbon chemical shifts. Carbon chemical shifts also will be useful for calculating the structure. Okay. So this is the analysis part of a large DNA sequence using heteronuclear experiments. So this is H1 prime, H3 prime to H1 prime, H3 prime NOE correlations between in the immuno correlations there. And this is, you see 12.5 ppm to 14.14 ppm. These are immuno correlations. Okay, this is H6, H8, H2 to H6, H8, H2. This is the base correlations. This is proton based. These are all proton based experiments. And similarly here also it is all, this also we discussed already. Battery is running low.
Yes, I used to difficulty.
So this is also proton distances here, 1 prime to 2 double prime, which I already showed you earlier. This is in the nosy spectrum. This is the base to H2 prime, H2 double prime region on the top of the portion of the on the whole spectrum which was indicated here. These are all individual assignments. And these are the immunoprotons, to the base protons. Immunoprotons are here. Okay, that this is gone.
Connection problem. एक्सटेंशन बोर्ड का प्रॉब्लम थोड़ा वायर हिल जाता है ना अच्छा ये बोर्ड का प्रॉब्लम
So these are from the immunoprotons. You see, this is 12.5 to 14 ppm. Okay, 12.5 to 14 ppm is the immunoprotons. Immunoprotons to the base protons. Those ones are here. The base pairs are indicated here. The immunoprotons, you will see base protons to its own base and to the next base. Okay, base, next base. So you will see two peaks therefore. G4 to C9. Okay, G4, C9, H4, H42. Okay, so therefore you will all of these peaks one has to analyze in the nosy spectrum. Now this is the spectrum of a intramolecular triplex. How does it form a triplex? Now you take this particular sequence here, indicated, where does it start from? The triplex patterns are indicated here. That's why I discussed all of these things in great detail with regard to the hydrogen bonding schemes. Okay, in the hydrogen bonding schemes, you have the bases are paired like this: T, A, T. AT is the Watson-Crick base pair, and GC is the Watson-Crick base pair here. And in the major group comes another T. This is from the third strand. Third strand pairs with this A here, like this, with this hydrogen bond. In this case, the C plus, the C gets protonated, and then it gets a hydrogen at the N3 position, and that pairs like this. Okay, this using this N3H and C4NH2, it pairs here like that with the GC on the G. So the T pairs on the A and the C plus pairs on the G base protons. Okay, now here is the sequence, which is the labeling is done in this manner here. This is they have used a particular funny labeling scheme here. It is the numbering scheme. The numbering scheme of this. They have 1, 2, 3, 4, 5, 6, 7. Then they have written here 8, 9, 10, 11, 12. This is not necessarily going along the 5' 3' sequence. They just label it from the point of view of analyzing here. So this is the particular duplex stem. This CC, TC, TTC, and G8AG, this stem here. Okay. The 5' sequence goes like this. The end of this, ideally one would have started saying, okay, 1 here and things like that. Okay, but they will represent in this manner to conveniently show the base pairing schemes there. Okay, so it goes like this. The DNA sequence goes like this, takes a loop, then comes like this, then takes a bigger loop, then comes like this and goes here. So therefore, which is the Watson-Crick base pair? This is the Watson-Crick base pair. Here, this first 7 to 8, this is the Watson-Crick pair. Why do I say Watson-Crick pair there? Because you see the third strand which is present here, this says C plus here, and the C plus there. The C plus can only happen in the case of the third strand. This is the third strand. Therefore, they have indicated there the strand forms a duplex like this. G from here it goes and then turns around. Then it forms a 7 here, and then it turns around like this and comes back here to pair with the individual G. See, the C plus pairs with the G. T is pairing with the A, that's like here. C plus is with the G. The T is pairing with A, okay, and C plus with the G. T with A, C plus with the G, C plus with the G. So these ones, TC has to get protonated. Now, if this happens, how many immunoprotons do I expect? I need to get a base proton immunoproton spectrum for all of these here, and that is what is indicated here. You see all of these spectra. This is for some variations, some modulations which have happened here. So you see C20. C20 is here. That comes C20 because it's protonated. The protonated C always comes very high, the immunoproton. That comes around 15 to 16 ppm. So on the basis of the chemical shift, you will identify that these ones belong to the protonated cytosine. Therefore, the 2018, these ones belong to the protonated cytosine. Then after that, you have the T's, T5, T3, T6, G4. So therefore, this is the way you can, these assignments that I've taken this from this particular paper there. They have calculated the full intramolecular DNA triplex. Intramolecular DNA, see this, look at the sequence here. Sequence starts from a G, that is here, right? It goes like this, GAA. Although in this they have written 8, they written that to identify the base pair. Okay, what kind of a base pair, numbering of the base pair. So this base pair G8 is here, this one is here. Although in the sequence this is actually number 1 here, but they have indicated that for the base pair, which way it is going. Anyway, it doesn't matter. Now, quadruplexes. This is a review NMR spectroscopy of G quadruplexes. How many different types of quadruplexes can be formed? G quadruplexes. Okay, these G quadruplexes are extremely stable, very stable structures. Okay, so here with the G tetrad, and you can form quadruplexes that indicated. This is a kind of a parallel stranded quadruplex and 3+1 hybrid. Okay, 3 plus 1 hybrid meaning 3 are going in one direction, 1 is going in the other direction. Anti-parallel here, there are 2 in one direction, 2 in opposite direction. Okay, you can see which 2 are in one direction. This and this are in one direction, these 2 are in the opposite direction. Okay, and they pair. Or anti-parallel, this is another way. You can have this and this are parallel, this and this are parallel. Here the 2 neighboring ones were parallel, and here the 2 diagonal ones are parallel. Okay, you can form a structure like that as well. All this depends upon what is the sequence of the molecule you have, and you can fold around in a different ways. Now here you see how this happens. Double chain reversal loop. So the chain starts here, then it has a loop here and comes back. Okay, and this is another one of the same, and then of course you form a quadruplex of this type. Here is edgewise loop. This is the edgewise loop, similar one happening on this side, and here is the diagonal loop. From here to here there is a loop. So this is a monomer within the same molecule. You have the multiple folding loop, multiple loops, and you can form a quadruplex structure, which is 2 G's stacked on each other. And this is the dimer. There are 2 molecules which are in opposite orientations, and they form the loop here and a loop there, and they can form a structure like this. So these are different kinds of quadruplexes which can be formed. And this is the NMR spectrum of such a kind of a within the same molecule quadruplex which is formed there. So the molecule is starting where? It is starting from starts from here. It is not written the 1 and 2. What they may not be necessary here. All these are the G's 3, 4, 5, and this is the loop area here. And then you start from 7, 8, 9. Once again a loop area. There is a big loop over there. Then you have 19, 20, 21, once again a loop, then 23, 24, 25. See, there are big loops in between, okay? And those ones go out and bring the G's in right juxtaposition, right positions to be able to form these quadruplexes. And the NMR spectra of such a thing is, yeah, we can see. If you have you can have a quadruplex which is going like this, the loop on the same side, Here the loops are on the opposite side. And in all of these cases, there will be variations in the glycosidic torsion angle. When you have these sort of different orientations of the strand, then you have different orientations of the loops. There will be changes in the glycosidic torsion angles. They can go from sin-NT complex. And here it is the sequence. The sequence is T, G4, T2, G4T. Okay, the T's obviously go into the loops, and it is the G4 and the G4 which form the pairs here. This is one molecule, this is another molecule. Two corresponding asymmetric dimeric G quadruplex topologies. 4 different strand conformations are colored, red, blue, green, and yellow. Okay, so all of these things can be present at the same time in your molecular system. Both the things can coexist. So therefore, your NMR spectra can get very complex. You have so many immunoproton peaks which are seen. All these belong to the G's only, remember. All these belong to the G's only. So these ones are all 2, 3, 4, 5, 6, 7, 8, 9. These are all the G's. Each G gives rise to 1. So therefore, all the individual G's are non-equivalent. Therefore, all of them produce separate peaks. 2, 3, 4, 5, 8, 9, 10, 11. This makes 8. And here again, you have 2 different 2, 3, 4, 5, 8, 9, 7, 10. 8, 9, 10, 11. See here. 2, 3, 4, 5, 8, 9, 10, 11. Then you see from all of these. So many of them you see. And all of them are different. The blues and the reds are different, and they're clearly color coded on this side here. So to analyze this is quite a job. Okay, now this is a very simple molecule which is easy to understand. Those ones are very complex situations. This is only Ag3T. Ag3T. There are 3 Gs and 1 A and 1 T. And so therefore, if it forms a quadruplex, how many Gs I should expect? If it's a symmetrical quadruplex, if it forms, I should get 3 Gs. So therefore, if I have a structure structure formed like that, which is the quadruplex, let us say, and you have for all the three G's are equivalent, three G's are different, but all the four strands are equivalent. Then you have three G peaks coming here, and G2, G3, G4, those ones are identified. Look at the stability of this small molecule. At 280 degrees, it is all of these are seen. These are the amino protons here present here, but the iminos are very stable, and they are stable up to 320 degree Kelvin. That shows the strength of the G quadruplex. This is in H2O solution, and these are the various base protons here, very sharp, and this is sharp up to 330 degrees. You can see a little bit of those, and then after that it melts. Then it is the immunoproton, the G quadruplex melts away. And the very unique thing which was observed in this particular situation was the ANH2. ANH2 is also stable. Normally you don't see the aminoprotons stable. Aminoprotons exchange out very rapidly. Even at a low temperature you don't see. But here the ANH2 is so stable up to 320 degree. So what does that mean? This means that actually aminoproton is also involved in kind of a tetrad formation. It is also involved in hydrogen bonding, and that was observed and to be true, and that led to a kind of a new structure wherein you not only have a G tetrad but also have a tetrad. All four, there are four A's which are in the same plane, and they form a tetrad configuration. So assignments of this spectrum is pretty simple here. The H8, H2, H8, H2 to the H1 prime, and all of these H1 prime correlations can be identified as I described to you before. You have the A1 can be easily identified. A1 shows only one peak in the H1 prime area because there is nothing above that. Then you see 1 to here, 2, 2 to 3, 3 to 4, 4 to 5. Very easy to identify. All the five are clearly identified. And then you see from A1 is at one position. You see a peak to the H2, from the base of H8 to H2. So you see A1 H8 to its own, and then you see there is A1 H2 peak. This is from the aminoproton to the H2 proton. Okay, that is an amazing thing. Aminoprotons are present here, and it also shows a peak to the H2 proton. Aminoproton here is showing to the H8 of A1, and is also showing a strong cross peak to the H2 proton of the same base. So this strong peak tells you that the aminoprotons is very stable, and it is hydrogen bonded, and it is very close distance to the H2 proton, and that is why you are seeing a strong cross peak over there. This actually led to the formulation of a new structure which is called as the A-tetride. And this was present in the Ag3T sequence. Okay. And this was not present, notice here, in the TAG3T sequence. The moment I put a T in the 5' end here, in the 5' end, Ag3T, these ones belong to Ag3T. And if I put a T at the 5' end, these ones disappear. It's no longer formula. So the tetrad is not formed if it is another T over there. So look at the variation, how that can happen. And this is Ag3T and this is TAG3T. And these are the aminoprotons to the various immunoprotons and so forth. This is more details, we will not go into that. And now you see the hydrogen bonding interactions. The A-tetrad, it can form, how does it involve the hydrogen aminoprotons in the formation of the hydrogen bond? The amino group is here. The amino group is here for all of these A's. The four A's you put together, see, and this one aminoproton, hydrogen bonds to the nitrogen here, and similarly it forms the nitrogen here, similarly to the nitrogen here. Okay, and this is the N1 position. It can form to the N1 position here. The aminoproton forms to the N1 position of A. Remember the sequence, numbering sequence which I showed you there. This goes 1, 2, 3, 4, 5, 6, 7, 8, 9. This is the N1 position. There is an alternative way of forming the hydrogen bonds, and that is indicated here. Here the aminoprotons hydrogen bond to the N7. So this is the N7 position. Okay. This is the N7 amino proton pair forms hydrogen bond to the N7 of the other one. So symmetrically they are all placed here. So this produces a larger hole here compared to the hole here. But still it is theoretically possible. So you can have both kinds of structures, NH2 to the N1 hydrogen bond and NH2 to N7 hydrogen bond. And these ones will of course cause variations in the structure of the molecule, and that is indicated here. This is the Ag3D structure there, and these are the stereo views of the structures. And these A and B are for the N6-1 and N6-7 models. N6-1 meaning amino to N1 position of A, and this is amino to N7 position A, the previous hydrogen bonding scenes which I showed you there. So these are two different possibilities. These are stereo pictures. This is the N6-1 model, this is the N6-7 model. And this is the structure of TAG3T. Now you see, when you have the TAG3T, the T at the top, the A becomes completely random. It is no longer a tetrad anymore. Look at this, this is so stable tetrad here. The structure is so well defined at this point. But here the structure is poorly defined, because the TA becomes a haphazard and it is very flexible and it doesn't form a stable structure there. Although the other ones, the G's are there all right, but the A tetrad is not seen clearly in this area. Okay, when after that we actually went forward to look at whether it is possible to form tetrads of other bases, whether the T tetrad can be formed. And indeed it turns out that you can also form a T tetrad. T tetrad was happening in the particular sequence of a telomere sequence there, and that is again that analysis came from the analysis of the NOESY spectra. The crucial thing was the T1H6 to T1 methyl there, and T4NH to T4 methyl. So all of these analyses which I described earlier could be identified there. Okay, so we will not go into the specific details, but the T tetrad has this sort of hydrogen bonding scheme. Hydrogen bonding scheme, what is the thing? There are two ways again here. See carbonyl of 1T. This is the carbonyl of 1T, and this is the NH. This is the NH, this is the NH proton, and this is the carbonyl. This is the oxygen. So NHO hydrogen bond there. And this is the O4 carbon. And this is the C5. O4 oxygen. Okay, this is the 4 position there. This is the 4 position. Okay, so here O4H3, then the T imino is T3 at the 3 position, O4H3, and here it is the O2H3. Notice there are 2 carbonyl groups in T. This is one carbonyl group and this is the other carbonyl group. Okay, now in this case it is this is 4 and this is 2, and here you see this is O4H3 hydrogen bond, here is O2H3 hydrogen bond. So you have to slightly rotate those groups, and then it will form a stable structure. So once again you form a very nice hole here in this position, and these actually can carry ions, potassium ions, sodium ions, and they can provide stability to that. The interior of this is very stable because it has very nice electrostatic atmosphere, and they can form a very stable structure. So this is the three-dimensional structure of this kind of a quadruplex with a T tetrad. Okay, this is the same with the space filling model. Okay. This is just a comparison of 2 different structures there. DNA sequences, both are quadruplexes. Both are quadruplexes. In one case, you have a G4 stretch. In this case, you have a TGG, TGGC. And here is TTGGGT, and determined in the presence of 100 millimolar KCl. The flattening of the backbone in the latter quadruplex can be seen. See, this quadruplex, the backbone is going like this. In this situation, the backbone is flattened here like that. Okay, that you can clearly see. So it becomes fatter. This becomes a fatter quadruplex, and that is in this case TTG4T. The structure is much fatter compared to this one. So this is comparison of the G quadruplex structures only. Okay, this is the summary of all the four A and T and C. We also discovered the C tetrad in one of the sequences. So that's a particular kind of a hydrogen bond scheme is also indicated there. In a 8-month DNA sequence, we could determine, we identify a C tetrad. And this is a T tetrad, a C tetrad, and two kinds of A tetrads there. We label this as the N61, N67 here, and you have the different kinds of tetrads formed. Okay, now this I already discussed, different kinds of what is the I motif I already showed in the previous slide. Now we come to the NMR of tRNA. The tRNA is a large molecule. A large molecule, it has of course the non-exchangeable protons in this area. The exchangeable immunoprotons are in this area. And all of these, you see, this is recorded at a very low temperature and then certain low pH, conditions there. Of course, I don't remember the exact conditions here. And all the immunoprotons are very clearly seen. You can identify the kind of a structure that may be formed. And once this is determined, identified, you can determine the distances from one immunoproton to the next immunoproton or to the sugar protons, and you can calculate the structure. And for all of this, you have to use now also heteronuclear NMR experiments. So homonuclear NMR alone is not going to be adequate there. We need to use the heteronuclear NMR experiments as well. Okay. Now, and for this, of course, you have to have the labeling. This is quite an expensive proposition. To label DNA or RNA is not easy. So there are various methods called in vitro transcription methods in vivo experiments which can do that is not easy. So, but anyway, people have labeled this by synthesized this using certain labeling procedures, and this is the nitrogen labeled RNA sequences. So you will see correlations with regard to the nitrogen. So what are the correlations seen there? This area is the immunoproton region, right? This is 11.5 ppm to the 14.5 ppm. This is the immunoprotons. The immunoprotons means they are attached to the nitrogen. Every immunoproton is attached to a nitrogen. Now, therefore, you have one bond correlation. If you do a HSQC experiment, heteronuclear correlation experiment, you will see correlation to its own nitrogen. Okay, you will see correlation to its own nitrogen, and depending upon the structure, you may say correlation to another nitrogen as well. So depending upon the structure, you will see that kind of a correlation. For example, here you see this U8A14N7. Okay, so U8N7, this is the pairing that has happened. U8 to A14, that is a pairing, that's the base pair. And it is showing a peak to its the N7 of A. N7 this is the purine, right? A is a purine, therefore it has the N7. And this is a cross peak is from the immunoproton of U to N7 of A. And this is immunoproton of U to its own nitrogen, N3. So U has a N3. So therefore there are two peaks. So in the base pair, you see a cross peak in, one has to remember the base pair structures, okay? So in the base pair itself, so if there are two hydrogen bonds, the immunoprotons is base pair to its own nitrogen, and then it also shows a correlation to the nitrogen of the other side, okay? So NHN hydrogen bond, So okay, let me say write this here, NHN hydrogen bond, right? So suppose this is the U and this is the A, okay? And which one is this? Let us say this is A, U8, this is A14. A14, this is the base pair. So this immunoproton shows a correlation to this and also shows a correlation to this. So you will see two peaks, and that is what is shown here. In each of these cases, you will see two peaks. This will allow you to identify the nitrogens of the individual base pairs. So therefore, you can figure out how the base pairing scheme is happening. in your DNA sequence or the RNA sequence. All of these now becomes very important in the case of very complex molecules. Otherwise, proton spectrum alone will not be able to tell you all of this, and you have all of these. The pairwise correlations which are seen here were extremely important to identify the structure of such large RNA molecules. RNA molecules are, especially the tRNA. tRNA is very, very complex there. And similarly, you see to the aminoprotons. Here, this is the iminoproton region once more. Iminoproton to the aminoproton. This is one hydrogen bond there, right? So, then you will see a hydrogen bond to the this is the NOE spectrum, okay, N15. To the in the another hydrogen, this thing is there. It will to the nitrogen of that one. You will see that correlation as well. N15 delta N15 ppm. Okay, G24C12. So therefore this requires quite a complex analysis here, and this is the imino correlations there. This is imino here, also imino here. So this will tell you how the bases are stacking here in this one base to the other base, and you will see correlations there. Imino proton cross peaks, meaning it is the stacking of the base pairs, and there you will see the imino cross peaks. And that becomes possible once you identify this on the basis of the heteronuclear experiments. Okay, I think that's the last.
Yeah, that will be last time.
This is the last. Yeah. Okay, good. All right. So I think we have covered the thing that had to be covered in time. That is okay. So any questions on these things? We can.
Can you just write?
Yeah.
The question, the doubt. This recording you are keeping, na? You record, but attendance. Attendance.
So I think with that we'll complete the nucleic acid part. Next time we will go over to proteins.
Any question? Anybody responded? No, sir. Okay, all right. I think no question.
Okay, good.
Then you can close it.
Now we can close. Yeah, sure.
Okay then.
Okay, so to the next one.
Just a minute. Participants, this our Professor Sudarshan will take a class on next Monday. Okay, one class he missed, so that class will be taken Monday, 3:30 to 4:30. Please attend. Okay, and Professor Hushru will take as usual program Wednesday and Friday at 3:30.